Hello, am I audible? The student who have joined, can you let me know if I am audible? And also please let me know if you can see the video. Thank you for confirming. Can you please let me know if you can see the presentation? Is the presentation visible to you? Please let me know. Okay. Thank you for confirming. We will wait till it is 6 p.m. and then we will start today's session. So kindly wait till then. Meanwhile, if you have any doubt or any query regarding the last class, please feel free to ask that questions. If there is any doubt, any problem regarding the last problem solving session, we can discuss it before starting the next session. We will start the session at 6 p.m. Let other students join, then we will start today's session. Okay. We will start the session within 5 minutes, so please wait. Let more students join, then we will start the session. Okay, I think I should start by introducing myself, or let me wait just for 1 minute, then I will start the session. Keep the Okay, so I hope my voice is audible. If not, please let me know in the chat box. And let us start the session. I hope more students will join within the due time. So let us start. If there is any issue with the audio or with the video, please let me know in the chat box. Okay. So it's 6 p.m. Let me start our today's week 4 live session for this NPTEL course of Organic Chemistry in Biology and Drug Development. So very good evening to the students who have joined. My name is Mousumi Deb. I am a PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAM course of Organic Chemistry in Biology and Drug Development. And today it is our week 4 live session. So the thing what we had do in our live session is like we discuss about the particular week's course content. Every week we have this kind of live sessions where we discuss that particular week's course content. Okay, and we discuss MCQ questions related to that particular week's topics, and all the questions will be will follow up with a detailed discussion, so that if the questions will be asked in a final examination in a descriptive manners, you can easily answer those questions. Now, before starting the session, so is there any doubt from the previous classes? I have asked this question before also. So if there is any doubt related to any before earlier sessions, you can also let me know. Okay. So today it is our week 4 live session. Week 4 course content was posted on your SWAM portal in the last Friday. Let us discuss about the particular week 4 course content in today's class. Before starting the session, I will show you where to find this problem solving sessions. Okay, let me just summarize few things, then I will discuss that particular point. So there will be a problem solving sessions every week, and these sessions are scheduled on every. Okay, so there is a mistake here. So the sessions which will have these sessions are on Friday from 8 to 6 p.m. Okay, and the sessions are live on Google Meet platform with the particular link. This is the link which you have used for joining in today's class also. This link will be reminded to you on every Thursday, I mean one day before the class. Okay, this will be reminded to you via email. Now, during the class, if there is any network disruption from my side, please do not leave the session immediately. I will try to join as soon as possible. Please remain in the class. Okay, and this I am recording the session. That is why I have muted your microphones and cameras, but the chat box is always open. We will use this chat box to communicate with you. Okay, every question, the answer, question answer, the doubts, everything will be communicated through the chat box, because I am recording the session. Now I want to record it without any problem. That is why I have to mute your microphones and cameras, but the chat box is always open, and I am monitoring the chat box every now and then. So if you have any doubt, any query, anything you want to tell me, you can let me know in the chat box. Okay, after class, if this doubt persists, you can drop an email to me. Okay, I will answer your queries via email also, or you can drop your queries in the discussion forum of the on the SWAM portal. That you can also do in email through via email. If I can solve your query, I will do it surely. Otherwise, I will discuss that your query in the next the upcoming class. Okay, this presentation which I am showing you today and the class recording, it will be shared to you after the class. So where you have to find these things. Okay, so what you have to do is you have to go to the SWAM portal. Okay, so you have to go to the SWAM portal on the course page, which is the Organic Chemistry in Biology and Drug Development. So there you will find all the announcement, all the class reminder and everything. You will find there is a left panel. Okay, the left panel will have a thing which is called problem solving session to July 2025. If you click on that and click on the recording portion, you will find something, a Google Sheet will open, and it will contain all the class recording, and also the drive, Google Drive link, which contains the presentations. Okay, the Google Drive, which looks like something like this, which will have the presentations. You can easily download, or you can view it from here also. Okay, and recording also, you will find the YouTube link of the particular class. That is also present in that web page of the SWAM. Okay, so today it is our week 4 live session. So like every live session, we will have around 20 MCQ questions. Each question will follow with 4 different options. I will, after showing you a particular question, I will wait for 2 to 3 minutes. Then I will check the chat box. Okay, After you try to answer the question, even if your answer is wrong, it is fine because there is no marks on answering, but there is a marking on the assignment which is present in the course page of the SWAYAM portal. Okay, so be careful while answering in that assignment because it has some mark, it carries some mark. So don't do any mistake in that assignment. But here in the class we don't have any marking, so you don't have to worry about anything. You can answer the questions during the session. Whatever you think is the correct answer, you can let me know through the chat box, because even if your answer is wrong, there is no problem. Okay, so now let us see what is the course content of today's class. So today's week 4 has these topics as a course content. We have enzyme kinetics, concept of enzyme inhibition, problems on enzyme kinetics and enzyme inhibition, synergy, synthetic biology. These are the topics. Okay. So in last class we have discussed about the secondary, tertiary, quaternary structure of protein. We have discussed about the purification techniques, the chromatographical techniques, whatever there is the chromatography, the gel electrophoresis. Okay, 2D electrophoresis, all these things we have discussed in our last class. Also we have discussed about the enzymes, what is the enzyme, what are the enzymes, how it is related. So if you look at the course pattern, first we have discussed about amino acids, we have discussed about the types of amino acids, what are the isoelectric points, how to solve the isoelectric points, those things, followed by the primary structure of the proteins and methods of the peptide bond synthesis, methods how to analyze what is the sequence of the amino acids in the peptide chain. That is what we have discussed in our second class, followed by our third class, which has lead to the enzyme and enzyme kinetics. So enzymes are very related to proteins because all the enzymes are a type of protein, and the enzyme kinetics is a very important and crucial chapter. In our last class we have discussed about the Michaelis-Menten enzyme kinetics. We have discussed how the enzymes behave and what is the kinetical graph looks like. We have discussed one or two mathematical questions also. Today's class we will focus on enzyme inhibition, mainly different types of enzyme inhibition, what are the type, how to identify what is the particular mode of inhibition. Okay, the graphs, how it relates with the enzyme inhibition. We will discuss a little bit about synthetic biology, and in today's class we will solve mathematical questions. Okay, so we will have 3 to 4 or more mathematical questions in today's class, which we will discuss thoroughly. These questions are very simple, just you have to understand the graphs and remember the equations a little bit. Okay, so let us start with the first question of today's session. I hope it is clear till now. If there is any problem or there is any doubt, please let me know. Okay, if there is any issue with the audio or with the video, anything, please let me know. Okay, so let's start. Let us look at the first question of today's session. So the first question of today's session is, which of the following diagrams best describes the equilibrium associated with a competitive inhibitor, where E represents enzyme, S represents substrate, and P represents product, I represents the inhibitor. Your first option looks like this: E plus S is in equilibrium with ES and is equilibrium with E plus P, and E plus S is E plus I is equilibrium with EI. Okay, so this represents the enzyme substrate complex. EI, it represents the enzyme inhibitor complex. Okay. Now option number B looks like this: E plus S is with equilibrium with ES, which is the enzyme substrate complex, which with I is equilibrium with EIS, and this is with equilibrium with E plus P. Then number C looks like this: E plus S is equilibrium with ES and E plus I is with EI. EI plus S is equilibrium with EIS, and ES plus I is also with equilibrium with EIS, and this is with equilibrium E plus P. And we have this also, where E plus S is equilibrium with inhibitor, and then which is equilibrium with ES, and this is in equilibrium with E plus P. So all the four are different equilibriums. Okay, so you have to identify which diagram best describes the equilibrium associated with a competitive inhibitor. Let me know in the chat box A, B, C or D which of the following is the correct answer. Which of the following diagrams best describes the equilibrium associated with a competitive inhibitor? I will give you 2 minutes, then I will check the chat box and we will discuss this question in detail. Which of the following diagrams best describes the equilibrium associated with a competitive inhibitor? So, which of the following diagram best describes the equilibrium associated with a competitive inhibitor? A, B, C, or D? Let me check the chat box. Let me check if there is any answer. Okay, I got one answer which is saying it is number B. Now let me check if number B is the correct answer or not. See, even if your answer is wrong, it does not matter. Please try to answer the questions, okay? So I have got one answer which says it is number B. Now let me check if it is the correct answer or not. So the equilibrium, the diagram which best describes the equilibrium associated with a competitive inhibitor will be number A. E plus S is equilibrium with ES, which is equilibrium with E plus P, and E plus I is equilibrium with EI, which is the enzyme inhibitor complex. Okay. So what is a competitive inhibitor? So basically, competitive inhibition of the enzymes. This diagram is it very accurately. It has describes what is a competitive inhibitor. So basically, competitive inhibitor has roughly the same shape as the substrate, so it can fit in the active site problem. Okay, the competitive inhibitor is able to fit at the active site. When it is there, the substrate is unable to bind with the enzyme, and the desired reaction does not occur. The inhibitor and the substrate both compete for the active site. The competitive inhibitor is often a product of the enzyme catalyzed reaction. When product is made, the product would compete with the substrate for the enzyme active site, showing the reaction down. So basically, competitive inhibitors are those inhibitors which has the size and shape almost roughly similar to the substrate, so it can easily fit with the in the active site of the particular enzyme. Okay, that is why you will see enzyme and substrate, these are is equilibrium with enzyme substrate complex, and also enzyme and inhibitor is equilibrium with enzyme inhibitor complex. Okay, which is in case of a competitive inhibitor. So normal enzyme, maximum rate of the reaction can be reached when the line plateaus is all enzymes are occupied with the substrate. This is a normal Michaelis-Menten I mean equivalent in case. Okay, competitive inhibitor. Remember, competitive inhibitor competes for the active site with the substrate. When the substrate concentration exceeds the inhibitor concentration, the reaction will proceed and the maximum rate of the reaction can be achieved. And non-competitive inhibitor, these inhibitors don't compete for the active site. They attach to an alternative site of the enzyme, changing the shape of the active site, preventing the substrate from binding. Non-competitive inhibition cannot be overcome by increasing the substrate concentration. Therefore, maximum rate of the reaction will not be achieved. Regardless of the substrate concentration, inhibition lowers the amount of the usable enzymes. Okay. So, in case of the competitive inhibition, one thing you can do is increasing substrate concentration. If the substrate is more, definitely the active site will be occupied by more substrate rather than the inhibitor molecules. Okay, so you can easily overcome the effect of the competitive inhibitor by increasing the substrate concentration, but that does not happen with the non-competitive inhibitor. The details about the non-competitive inhibitor we will discuss in our upcoming slides. Okay, so is it clear or is there any doubt? The first question, is it clear? I hope it is clear. If you have any doubt or any query, please let me know after every question if you have any doubt or any query regarding the particular question. Okay. So this is the equilibrium of competitive inhibition, and this is about the competitive, how the competitive inhibition happens. Okay. Let us go to the next problem. Problem number 2 says, which of the following inhibitions occurs in multimeric enzymes? Your options are competitive inhibition, non-competitive inhibition, uncompetitive inhibition, and irreversible inhibition. Which of the following inhibitions occur in the multimeric enzyme? Competitive, non-competitive, uncompetitive, or irreversible inhibition? Which of the following inhibitions occurs in multimeric enzymes? Let me know A, B, C or D, which of the following you think is the correct answer for problem number 2. Which of the following inhibition occurs in the multimeric enzymes? Competitive, non-competitive, uncompetitive or irreversible? I will wait for 1 or 2 minutes, then I will check the chat box. In multimeric enzymes, which kind of inhibition you can find? Competitive, non-competitive, uncompetitive or irreversible? Let me check the chat box. Okay, I got one answer which is saying it is number D. Let me check if number D is the correct answer for the problem number 2. Okay, so the inhibition that occurs in multimeric enzyme will be, it is number C, uncompetitive inhibition. Okay, now let us understand what is uncompetitive inhibition. So basically the uncompetitive inhibitor has no structural similarity with the substrate. Okay, it may bind to the free enzyme or it may bind with the enzyme substrate complex that exposes that the inhibitor binding site. Its binding, although away from the active site, causes the structural distortion of the active and allosteric sites of the complex enzyme that inactivates the catalysis. This leads to the decrease of both Km and Vmax. Okay, Km is the Michaelis-Menten constant and Vmax is the maximum rate of the enzyme catalyzed reaction. We have discussed this thing in the previous class. Okay, this inhibition cannot be reversed by increasing the substrate toward the saturating concentration, or reversal requires special treatment like dialysis. This type of inhibition is rare and is encountered in multi-substrate enzymes where the inhibitor competes with one substrate to which it has structural similarity and it is uncompetitive to the other. Thus, a reaction without the inhibitor will be E plus S1 is ES1 plus S2 is ES1S2 equal to E plus P. Okay, but with the uncompetitive inhibitor, it becomes like this. Okay, this is the uncompetitive where like E plus S1 equal to ES1 plus I is ES1I. It prevents the S2 binding and produces, and no product is there. Okay, this steady state kinetics for the uncompetitive inhibition is like this. Okay, so in case of multi-substrate, if the enzyme binds with one enzyme substrate complex and then it hinders the binding of the next substrate. Okay, now like this. So this is very important. This I will be highlighting this equation. So this is the normal equation without any inhibitor. Okay, without any inhibitor, this is the equation, and in presence of the inhibitor, the steady state kinetics is as follows. Okay, which is this one. So this is the other thing. This is the steady state kinetics. What is the examples? Uncompetitive reversal inhibitions is rare, may occur in multimeric enzymes. Some examples of uncompetitive reversal inhibition is the key regulatory heme synthetic enzyme. Okay, the delta amino levulinate synthesis and dehydrates and heme synthesis by heavy metal ion, that is lead, Heavy metals like lead from the mercaptides with the thiol act as the, at the active site of the enzyme. Okay, this acts. So this is the reaction. Oxidizing agents like ferricyanide also oxidizes thiol to disulfide linkage. The ability of the lithium to exert the profound and selective psychopharmacological effects has been focused as a considerable research effort. There is increasing evidence that lithium exerts the therapeutic action by interfering with the polyphosphoninositide metabolism in brain and prevent the inositol recycling by the uncompetitive inhibition of inositol monophosphate. Few pesticides are uncompetitive inhibitors. The best known example is the herbicide N-phosphomethylglycine or commonly known as the glyphosate or Roundup, an uncompetitive inhibitor 3-phosphoschemate 1-carboxyvinyl transferase. Okay, so these are the examples of the uncompetitive inhibition. So is there any doubt? If there is any doubt, please let me know. This was our second problem of today's session. This is all about the uncompetitive inhibition. In our last, we have discussed about the competitive inhibition, and this is about the uncompetitive inhibition. Okay. One by one, we will discuss different type of inhibitions. Okay. Now, problem number 3. It says that what is the mechanism of action of the cyanide poisons? Your options are number A, competitive inhibition, number B, non-competitive inhibition, number C, uncompetitive inhibition, number D, irreversible inhibition. What is the mechanism of action of the cyanide poisons? Competitive inhibition, non-competitive inhibition, uncompetitive inhibitions, or irreversible inhibition? Let me know in the chat box A, B, C or D which of the following you think is the correct answer. What is the mechanism of action for the cyanide poisons? Your options are number A, competitive inhibition, number B, non-competitive inhibition, number C, uncompetitive inhibition, number D, irreversible inhibition. Even if your answer is wrong, does not matter, but please try to answer the question. Let me know in the chat box A, B, C or D which of the following you think is the correct answer. What is the mechanism of action of cyanide poisons? What is the mechanism of actions of the cyanide poison? Let me check the chat box. Let me check if there are any answer for the problem. Okay, I got one answer, number D. Others? Anyone else who wants to answer this problem? See, there is no negative marking for answering during the question during the problem solving session, so you can answer. Even if your answer is wrong, it's fine. I got only one answer which says it is number D. Okay, let us check what is the correct answer for the problem number 3 and then let us discuss about it. So, the correct answer will be, it is number B, non-competitive inhibition. Okay, now let us discuss what is non-competitive inhibition. So, non-competitive inhibitor does not have any structural similarity to the substrate, but it binds both the free enzyme and the enzyme substrate complex. So, this is the steady state equation of the non-competitive inhibition. So, if you remember in our first question, we have discussed what is the steady state equation for the competitive inhibition. Here I have mentioned what is the steady state equation of the uncompetitive inhibition, and this is the steady state equation for the Non-competitive inhibition. So remembering the steady state equations are very important. Okay, this steady state equations will help you to understand the particular inhibition better, and also it will help you to question the mathematical problem, answer the mathematical problems. Okay, let me know if you do not understand the steady state equation. I will repeat one more time. So for non-competitive inhibition, this is the steady state equation where the substrate and enzymes are in equilibrium with the enzyme substrate complex. The free enzyme is in equilibrium with the enzyme inhibitor complex. Also, the enzyme substrate complex and enzyme inhibitor complex both are in equilibrium with the enzyme substrate inhibitor complex. Okay, and the enzyme substrate complex is the only thing that gives us the enzyme plus product. So the presence of the substrate has no influence on the ability of a non-competitive inhibitor to bind an enzyme and vice versa. Its binding alters the conformation of the enzyme and reduces its catalytic activity due to the changes in the nature of the catalytic groups at the active site. Enzyme inhibitor and substrate inhibitor complexes are non-productive. On increasing the substrate to the saturating concentration does not reverse the inhibition, leading to the unaltered Km but reduced Vmax. Okay, this is very important. We will discuss it in later also, how the Km and Vmax are affected in each and every kind of the inhibition. Okay, reversal of the inhibition requires a special treatment like dialysis or pH adjustment. Examples of the non-competitive inhibitors are mostly the poisons. Cyanide and azides inhibited the enzymes with the iron and copper as the component of the active site and prosthetic group, that is cytochrome C oxidase. Okay, for example, cyanide binds with the iron 3 of the in the heme in the cytochrome AA3 component of the cytochrome C oxidase and prevent the electron transport to oxygen. Mitochondrial respiration and energy production cease and the cell death rapidly occurs. The central nervous system in the primary target of the cyanide toxicity. Acute inhalation of the high concentration of cyanide, like smoke inhalation during a fire or automobile exhaust, provokes a brief central nervous system stimulation, rapidly following by conversion, coma, and death. Acute exposure to the lower amounts can cause the lightheadedness, breathlessness, dizziness, numbness, and headaches. Cyanide is present in air as hydrogen cyanide, in soil and water as cyanide salts, and in foods like cyanoglycosides. Okay, so the cyanide poisons are the very good example for the non-competitive inhibition. So in any kind of inhibition, what you have to remember is what is the inhibition, I mean how it happens. Okay, then you have to understand what is the steady state equation for that particular inhibition, then followed by the example of the particular inhibition. So these are the things which you need to understand in a particular inhibition. Okay, I hope the problem number 3 is clear to all, or should I repeat anything? Please let me know. Okay, now I will go to the next problem. The problem number 4, it says that what is the mechanism of action of orlistat, an anti-obesity drug? Your options are: number A, competitive inhibition; number B, non-competitive inhibition; number C, uncompetitive inhibition; and number D, irreversible inhibition. What is the mechanism of action for orlistat? Orlistat is an anti-obesity drug, and what is the mechanism of action of it? Competitive inhibition, non-competitive inhibition, uncompetitive inhibition, or irreversible inhibition? A, B, C, or D? Let me know in the chat box which of the following will be the correct answer. What is the mechanism of action of orlistat, an anti-obesity drug? Competitive inhibition, non-competitive, uncompetitive, or irreversible inhibition? Let me check the chat box. Okay, I got one answer, number D. Let me check if number D is the correct answer or not. So, the mechanism of action. Okay, this was the number 3, question number. Okay. So, the mechanism of action of orlistat, which is an anti-obesity drug, will be, it is number D, irreversible inhibition. Okay. No, so irreversible inhibition for the treatment of obesity. Fat in the diet is composed mainly of the triglycerides, which are digested in a small intestine to fatty acids and two monoglycerides. Now, the digestion products are then absorbed and act as the building blocks of the fat biosynthesis of our body. The enzyme pancreatic lipase is responsible for catalyzing the digestion of fat. So, inhibition of this enzyme will result in the reduced absorption of the glycerides and fatty acids from the gut. Consequently, lesser fat will be synthesized in the body. Orlistat is an anti-obesity drug that acts as an irreversible inhibitor of the pancreatic lipase as a result of the presence of the an electrophilic four-member lactone group. It acylates a serine residue on the active site, which is a part of the catalytic triad of the serine, histidine, and aspartic acid. Okay, so this is the pancreatic lipase, which is the enzyme which is responsible for the catalyzing the digestion of the fat. We have to inhibit this enzyme for the lesser consumption or the lesser absorption of the glycerides and fatty acids from the gut. So in the active site of this particular enzyme, we have the catalytic triad of serine, histidine, and aspartic acid. Serine we know has a hydroxyl group. Okay, like this hydroxyl, this OH group acts as a nucleophile, attacks on this four-member lactone ring for the orlistat, which is the particular anti-obesity drug, and it acts as an irreversible inhibitor because of the formation of this. Okay. So this is the mechanism how it happens. So irreversible enzyme inhibitors can form a covalent bond with a key amino acid of the active site and permanently block the affected enzyme. Okay, so this is very important, which you have to remember. Now, what is the irreversible enzyme? So an irreversible enzyme inhibitors, what does this inhibitors do? These inhibitors make form an covalent bond. Okay, they form an covalent bond permanently with the amino active site in the with the amino acid present in the active site of the enzyme, and it blocks the affected enzyme. The most effective irreversible inhibitors are those that contain an electrophilic functional group X capable of reacting with a nucleophilic group present in the amino acid side chain. Invariably, the amino acid affected is either serine or cysteine because these amino acids are often present in the active sites and contain nucleophilic OH and SH group respectively that are involved in the mechanism of many enzyme catalyzed reaction. Electrophilic functional groups used in the irreversible inhibitors include alkyl halides, epoxides, alpha beta unsaturated ketones, or strained lactones or lactams. The highly toxic nerve agents contain electrophilic fluorophosphonate groups and are irreversible inhibitor of the mammalian enzymes. Irreversible enzyme inhibitors are not competitive inhibitors. The increasing the concentration of the substrate will not reverse their inhibition as the inhibitor cannot be displaced from the active site. This can cause problems if the buildup of the particular substrate leads to the toxic side effects. For example, the monoamine oxidase inhibitors are block the metabolism of the noradrenaline and have antidepressant activity. Unfortunately, the metabolism of the substrate other than the noradrenaline is also inhibited, leading to the buildup of these compounds and a serious side effects. More modern MIOIs are designed and reversible inhibitor in order to avoid this problem. Okay. So this is all about the irreversible inhibition, how it happens, what is in, and what is the example. Again, is there any doubt? If there is any doubt, please let me know. Okay. So yes, number D was the correct answer, irreversible inhibition. Very good. Now let us go to the next problem. Okay. The problem number 5, it says that which kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A? Your options are number A reversible, number B allosteric, number C suicide, and number D irreversible. Which kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A? Reversible, allosteric, suicide, or irreversible? Let me know A, B, C, or D which of the following is the correct answer for problem number 5. What kind of inhibitor glucose 1-phosphate is for enzyme phosphorylase A? Reversible, allosteric, suicide, or irreversible? I will wait for another minute, then I will check the chat box. What kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A? Reversible, allosteric, suicide, or irreversible? Let me check the chat box now. Okay, I got one answer, number B. Let me check if number B is the correct answer or not. Okay, the kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A will be number B, allosteric inhibitor. So, what is an allosteric inhibitor? Virtually all enzymes are controlled by the agent which can either enhance or inhibit their catalytic activity. Such an control affects the local conditions within the cell. For example, the enzyme phosphorylase A catalyzes the breakdown of the glycogen, okay, a polymer of glucose monomers to glucose 1-phosphate subunits. This is stimulated by the adenosine 5' monophosphate or AMP and is inhibited by the glucose 1-phosphate. Thus, rising the level of the product glucose 1-phosphate will act as a self-regulating brake on the enzyme. How does its control takes place? The answer is that many enzymes have binding site which is separate from the active site, which is known as the allosteric binding site. This is where the agents controlling the activity of the enzyme bind. When this occurs, an induced fit takes place which alters not only the allosteric site but also the active site. Agents that inhibit the enzyme produce an induced effect that makes the active site unrecognizable for the substrate. Okay, so this is the glycogen and this is the glucose 1-phosphate, the product of this particular catalysis. Okay, and the phosphoroglucose and this phosphorylase A is the enzyme which inhibits this enzyme, and AMP which actually catalyzes this particular it stimulates the AMP stimulates this enzyme, and glucose 1-phosphate inhibits this enzyme. Okay, so and glucose 1-phosphate is the allosteric inhibitor, and this is the visual representation how the allosteric inhibitors work. This is the enzyme, this is the active site, this is the substrate, and allosteric inhibitor is this. As you can see, the substrate, it will fit very properly and very accurately with the in the active site of the enzyme. But if the allosteric inhibitor it binds with the enzyme, what happens? The conformational changes happen in the active site, and active site gets shut, and thus now the substrate cannot bind. okay, because it does not fit in the this altered active site. So this is the example of the allosteric inhibition, and this is this glucose 1 phosphate and A and phosphorylates A are the very good example of the how this allosteric inhibitors work. Okay, so yeah, number B was the correct answer. Now I will go to the next problem. Problem number 6 says, in case of the uncompetitive inhibition, which of the following is correct? Number A, Km is increased, Vmax is unaffected. Number B, Km is increased, Vmax is reduced. Number C, Km is reduced, Vmax is reduced. Number D, Km unaffected and Vmax is increased. In case of uncompetitive inhibition, which of the following is correct? Number A, Km increased, Vmax unaffected. Number B, Km increased, Vmax reduced. Number C, Km reduced, Vmax reduced. Number D, Km unaffected, Vmax increased. In case of uncompetitive inhibition, which of the following is correct? Number A, Km is increased, Vmax is unaffected. Number B, Km is increased, Vmax is reduced. Number C, Km reduced, Vmax reduced. And number D, Km unaffected, Vmax increased. Let me know in the chat box A, B, C or D which of the following will be the correct answer for problem number 6. Let me check the chat box. Let me check if there are any answer for this problem number 6. Okay, I got one answer, number B. Let me check if number B is the correct answer or not. Okay. So in case of uncompetitive inhibition, the correct option will be, it is number C. Okay, sorry, there was a mistake in this answer. Okay, let me just correct it out. Okay, the option will be C. I just have the make mistake in writing in case of the answers. Okay, let me just correct it out. Yeah. So the correct answer will be number C. Km and Vmax both are reduced in case of the uncompetitive inhibition. Okay. I will discuss it. So just let me correct it. So what happen in different kinds of inhibition? Now this is very important thing. Okay, this particular graphs, these are the Lineweaver Burke plots of the inhibition. So these are very important. Okay, so in case in the last class I have discussed about the Lineweaver Burke plot, okay, which is the double reciprocal plot also we know, which comes from the making the reciprocal of the Michaelis-Menten equation. It is very easy. I will not discuss today how to derive this Lineweaver Burke plots and how to derive these plots and what is the way. You can check in the last week's PPT. Okay, then you will find. So these are the Lineweaver Burke plots for the enzyme in different type of inhibition. Now you have to look at the things very careful. The red lines they implement indicates the presence of the inhibitor, and the blue lines are the in the without the inhibitors. Okay, so in case of the competitive inhibition, as you can see how the graph look like. So the both the things, the both the straight lines, okay, they cut the y-axis on a same point. Okay, now in case of the Lineweaver Burke plot, the slope is the k by Vmax. Okay, slope, I will just write it here. Slope equal to km How the slope is km by Vmax, and why is the intercept is the, let me write the intercept, and why the intercept is 1 by Vmax. These things I have discussed in our last class. Okay, so I will not discuss it today. You can follow that particular PPT. Just you have to remember that the slope for the Lineweaver bark plot is km by Vmax and the intercept is 1 by Vmax. Okay. So in case of the competitive inhibition, as you can see, they both are the lines, the presence of inhibitor and in the without the inhibitor, the graphs they cut on the same point on y-axis, means the Vmax 1 by the slope is same. Okay, this particular enzyme inhibitor does not alter the slope, but it alters the particular it does not alter the intercept, but it alters the slope of the particular graph. Okay, so if the 1 by Vmax remains unaltered, so what will happen? So the 1 by Vmax is unaffected. So Vmax will remain unaffected. Okay, if the V but the slope is changed. Okay, as you can see, the slope is higher in case of the with inhibition rather than the without inhibition. So since the slope is higher, the slope is Km by Vmax. If Vmax is not changed and slope is increasing, that directly means that Km is increasing. So as you can see, Km is increased and Vmax is unaffected. This is in the case of the competitive inhibition. In case of the uncompetitive inhibition, what is happening? You can see the slopes are the same. Now the slopes are the same. Okay, but the intercept is now in changed. Okay, so if 1 by Vmax in presence of inhibitor is greater than 1 by Vmax, let me say it is in presence of inhibitor. Okay, now is greater than 1 by Vmax without inhibitor. Without inhibitor, let me write it. Okay, now so if the without inhibitor it happens like this, then automatically it implies that Vmax in presence of inhibitor will be less than Vmax in presence of in absence of inhibitor. Without inhibitor, that means Vmax is reducing. Okay, and since the slope is same, Vmax is reducing. It indicates that Km will also have to reduce to make the slope or unchanged. Okay, to keep the slope unchanged. So that is the case of the uncompetitive. And in case of non-competitive inhibition, the Km remains unaffected, but the Vmax is reduced. If the Vmax is so as you can see, if you just extrapolate this thing, so on this thing, on the negative x-axis, it cuts on a same point, which is the minus 1 by Km. Okay, since it is cutting on the same point, which is minus 1 by Km, it indicates that Km remains unaffected, and since Km remains unaffected, the Vmax will reduce because 1 by Vmax, as you can see, it is similar than this. The Vmax is reduced, but Km is unaffected. So these are the thing for the these are the Lineweaver bark plots for the this is for in case of the uncompetitive. Okay. So this is how it happens in case of the uncompetitive, non-competitive and uncompetitive inhibition. This is very, very important thing. Okay, this is I will just make a star here because this Lineweaver bark plots with different kinds of inhibitions are a very important topic which you need to remember or which you need to understand. So is it clear or do I need to repeat it? If I have to repeat it, please let me know. Otherwise, I will proceed to the next problem. It is very important and it is not so hard. It is not very much hard actually. It is actually easy, but you have to practice it 1 or 2 times by yourself. Okay. I will go to the next problem if there is any is no doubt. Okay. Problem number 7, it says, which of the following inhibition is called the mechanism-based enzyme inhibition? Your options are number A, competitive inhibition, number B, non-competitive inhibition, number C, suicide inhibition, number D, active site-directed irreversible inhibition. Which of the following inhibition is called the mechanism-based enzyme inhibition? Competitive, non-competitive, suicide or active site-directed irreversible inhibition? Which of the following inhibition is called mechanism-based enzyme inhibition? Competitive, non-competitive, suicide or active site directed irreversible inhibition? Which of the following inhibition is called the mechanism-based enzyme inhibition? Competitive, non-competitive, suicide inhibition, or active site-directed irreversible inhibition? I will wait for 2 minutes, then I will check the chat box. Let me know till then which of the following enzyme is called mechanism-based enzyme inhibition: competitive, non-competitive, suicide, or active site-directed irreversible inhibition. Which of the following inhibition is called mechanism-based enzyme inhibition? Let me check the chat box. Okay, I got one answer, number D. Let me check if number D is the correct answer or not. So the inhibition which is known as mechanism based enzyme inhibition is, it is number C, suicide inhibition. Okay. So what is suicide inhibition? In suicide inhibition of a suicide inhibitor, initially inactive, attached with the active site of the enzyme, undergoes the catalysis to form the product, and then it occupies the active site, covalently binds to it. This binding alters the enzyme in such a way that the enzyme is damaged irreversibly. Okay, so this is the suicide inhibitor. It first attached with the active site of the enzyme, then it undergoes the catalysis and forms a product which will now form a covalent bond with the active site of the enzyme active site, and this enzyme product complex now will damage the enzyme forever. So that is why it is known as the suicide inhibition. So it is also known as the mechanism based inactivation. Okay, the inhibitor makes the use of enzyme's own reaction mechanism to inactivate it. In suicide inhibition, the structural analog is converted to a more effective inhibitor with the help of the enzyme to be inhibited. For example, ornithine decarboxylase catalyzes the conversion of the ornithine to putrescine, which is necessary for the polyamine synthesis. When the ODC in trypsinoma is inhibited, the multiplication of the parasite is arrested. Therefore, it inhibits the ODC enzymes such as the difluoromethyl ornithine have been found effective against the trypanosomiasis, which is the sleeping sickness disease. A similar mechanism is observed in case of the allopurinol, which is oxidized by the xanthine oxidase to alloxanthine, that is the strong inhibitor of the xanthine oxidase. The anti-inflammatory action of the aspirin is also based on the suicide inhibition. The arachidonic acid is converted to the prostaglandin by the enzyme cyclooxygenase. Aspirin catalyzes a serine residue in the active center of the cyclooxygenase. Thus, prostaglandin synthesis is inhibited, and so inflammation subsides. Okay. So this is the example, and these are the examples of suicide inhibition, and this is about what is the suicide inhibition and how it happens. Okay. Now I will go to the next problem. Problem number 8, it says that what is the catalytic triad of the amino acids in the active site of the enzyme chymotrypsin? Your options are number A, serine, arginine, glutamic acid, or glutamate residue, and number B, threonine, histidine, methionine, number C, serine, histidine, aspartic acid, and number D, threonine, arginine, and glutamic acid. Where what is the catalytic triad of amino acids present in the active site of the enzyme chymotrypsin? Option number A, serine, arginine, glutamic acid. Number B, threonine, histidine, methionine. Number C, serine, histidine, aspartic acid. And number D, threonine, arginine, and glutamic acid. Think and let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 8. What is the catalytic triad of amino acid present in the active site of the enzyme chymotrypsin? Serine, arginine, glutamate as or threonine, histidine, methionine, serine, histamine, aspartate, and threonine, arginine, glutamate. Which of the following will be the correct answer for problem number 8? Let me check the chat box. So what is the answer for problem number 8? Number B I got as the answer. Let me check if number B is the correct answer or not. So the catalytic triad of amino acid present in the active site of the enzyme chymotrypsin will be, it is number C, serine, histidine and aspartate. Okay. So this is the hydrolysis of the peptide bond catalyzed by the chymotrypsin. I think I have discussed it in our second class, how what the chymotrypsin does, but I had not discussed how the mechanism happens. Okay, because I had to discuss it in today's class. So the amino acids serine, cysteine, okay. So yeah. The amino acids serine, cysteine are present in some active site enzymes. These amino acids have the nucleophilic OH and SH group respectively, which are able to participate in the reaction mechanism. They do this by reacting with the substrate to form an intermediate that would not be formed in the uncatalyzed reaction. These intermediates offer an alternative reaction pathway which may avoid a high energy transition state, hence increase the rate of the reaction. Normally, an alcoholic OH group such as on the serine is not a good electrophile. However, there is usually a histidine residue close by to catalyze the reaction. For example, mechanism by chymotrypsin hydrolyzes the peptide bonds involve the catalytic triad of the amino acid, serine, histidine and aspartic acid. Serine and histidine participate in the mechanism as the nucleophile and acid-base catalyst respectively. The aspartic group interacts with the histidine ring and serves as the to activate and orient it correctly for the mechanism. The presence of the nucleophilic center, nucleophilic serine residue means the water is not required in the initial stages of the mechanism. This is important firstly because water is poor nucleophile and may also find it difficult to penetrate in the occupied active site. Secondly, a water molecule would have to drift into the active sites and search out for the carbonyl group before it would attract it. This would be sometimes similar to the game of the blind man's bluff. The enzyme, however, can provide a serine OH group as positioned exactly in the right spot to react with the substrate. Therefore, the nucleophile has no need to search for the substrate is delivered to it. Water is eventually required to hydrolyze the acyl group attached to the serine residue. However, this is much easier step for hydrolysis the peptide link as the esters are more active than amides. Furthermore, hydrolysis of the peptide link means one half of the peptide can drift away from the active site, leaving room for water molecule to enter. So, a similar enzymatic mechanism involved in the enzyme acetylcholine esterase, pancreatic lipase, and a viral protease enzyme carried by hepatitis C virus. Okay, so let us understand the mechanism now. This is my enzyme. Okay, this is the serine, histidine and aspartate which is present in as a catalytic triad. Okay, serine acts as a nucleophile, histidine acts as an acid-based catalyst and aspartate it activates the histidine and orients it properly. So first step is the activation of this histidine by the aspartate and activation of serine by the histidine. After the histidine it activates the particular hydroxyl group of the serine. Then what happens? It forms a bond with the carbonyl carbon of the peptide linkage, and then acid base catalysis happen, the proton transfers happen, and in the later stages for when this the after the so for dissociation of this particular group and regeneration of regeneration of this enzyme, water comes and acts as a nucleophile to hydrolyze this particular group. Okay, so give the for regeneration of the serine hydroxyl group. Okay, so this is how the catalysis or hydrolysis of the peptide bond happens by the enzyme chymotrypsin, and this is how this particular this thing happens. Okay, and this is a very good example of how this thing the different amino acids they behave in the active site and for catalyzing a particular reaction. Okay, so remember these are the amino acid present in the chymotrypsin active site, and this is the mechanism. This is a very easy mechanism, just you have to draw it in your copy and practice it 1 or 2 times. Now let me go to the next problem. Problem number 9 says inhibition of cytochrome P450 by tyalinic acid is an example of dash inhibition. Your options are number A competitive inhibition, number B non-competitive inhibition, number C suicide inhibition, number D active site directed irreversible inhibition. Inhibition of cytochrome P450 by tyalinic acid is an example of which kind of inhibition? Competitive, non-competitive, suicide or active site directed irreversible inhibition. Let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 9. What kind of inhibition is this? The inhibition of cytochrome P450 by tyalinic acid. Inhibition of cytochrome P450 by tyalinic acid is an example of dash inhibition. Competitive, non-competitive, suicide or active site directed irreversible inhibition. Inhibition of cytochrome P450 by tyalinic acid. What is this kind of inhibition? Let me check the chat box. Okay, I don't have any answer in the chat box. Okay, let me check what will be the correct answer and let us discuss it a little bit. So the inhibition of cytochrome P450 by tyalinic acid is an example of suicide inhibition. So suicide substrates are the agent which are converted to highly reactive species when they undergo the enzyme catalyzed reaction. They form covalent bonds with the enzyme and inhibit it irreversibly. In some cases, it can cause toxicity. For example, the diuretic agent tyalinic acid had to be withdrawn from the market because it was found to act as a suicide substrate for the cytochrome P450 enzyme involved in the drug metabolism. Unfortunately, the metabolic reaction carried out by these enzymes converted tyalinic acid to the thiophene sulfoxide, which is proved to be highly electrophilic. This encouraged a Michael reaction leading to the alkylation of the thiol group in the enzyme's active site. Loss of water from the thiophene sulfoxide restored the thiophene ring and resulted in the formation of the covalent link to the enzyme, thus inhibiting the enzyme irreversibly. Okay, now let us see how this particular mechanism goes on. So this is the example of irreversible or suicide inhibition of the cytochrome P450 by tyalinic acid. This is the cytochrome P450 which has this thiol group in its active site. This is the structure of the tyalinic acid. Okay, so the first step is the oxidation, and but this oxidation gives rise to this thiophene sulfoxide. Okay, the thiophene sulfoxide which it acts as a suicide substrate, and now the thiol group of this cytochrome P450 it attacks on this group this carbon C2 on the this position of the thiophene group, and this type of bond rotation happens, and which give undergo now the thiol group has undergone the alkylation. This and then proton transfer on these things forms this compound which is the enzyme alkylated and inhibited. Okay, so this is the enzyme alkylated and inhibited. So this is the example, a very good example actually, and this is a very good example how the drugs are withdrawn from the market after it is used for some time. It is also known as the post-market surveillance, where it is found. Okay, sometimes when we test the drug on a couple of number of people during the clinical trial, it may not show the side effects, but when it is in market and it is tested on a large number of people, it might show this kind of side effects. Then the drug will be withdrawn from the market. Okay. So tylenic acid is such an example, and the mechanism is very simple, but you have to practice it 1 or 2 times. Now I will go to the next problem. Problem number 10. Which of the following statements is correct? Your options are: number A, epitopes occur on antibodies. Number B, epitopes are known as antigenic determinant. Number C, paratopes are known as antigenic determinant. Number D, all of the above. Which of the following statement is correct? Again, I will repeat. Number A, epitopes occur in antibodies. Number B, epitopes are known as antigenic determinant. Number C, paratopes are known as antigenic determinant. And number D, all of the above. Let me know A, B, C or D, which of the following is the correct answer for problem number 10. Which of the following statements is correct? Epitopes occur on antibodies. Number A, number B, epitopes are known as antigenic determinant. Number C, paratopes are known as antigenic determinant. Number D, all of the above. Let me check the chat box now. Okay, I got one answer which is saying it is number D. Now let me check if number D is the correct answer or not. So the statement which is correct will be, it is number B. Epitopes are known as antigenic determinant. This statement is true. Now, what is an epitope? An epitope, also known as antigenic determinant, is a part of the antigen and is recognized by the immune system, specifically by antibodies, B cells and T cell. And paratopes is the part of the antibody which recognizes an antigen, the antigen binding site of the antibody. It is a small region of 15 to 22 amino acid of the antibody's FV region and contains parts of the antibody's heavy and light chains. So this is the antibody and this is the antigen. Antigen, it contains the epitopes, and antibodies, they contains the paratopes. Okay, and hapten, we also have something called hapten, which is a small molecule that is antigenic but non-immunogenic, and immunogen is the substrate. Okay, so these are the things related to our synthetic biology portion. So what are the differences between the epitope and paratope? Epitope, the part of an antigen molecule to which an antibody attached itself. Paratope, the part of the molecule of an antibody that binds to an antigen, aka antigenic determinant. It is also known, the epitopes are known as antigenic determinant, paratopes are known as the antigen binding site. Epitope occurs in both self and non-self antigens. Paratope occurs in antibodies. Epitope binds to the paratope on the antibodies and receptors on both B and T cells, and paratope binds on the epitope. Epitopes helps to discriminate between the self and non-self, and a non-self, and paratope helps to recognize the specific antigens and trigger a humoral immune response. Okay, so these are about the antigen and antibody. So the statements, first statement, it says that epitopes occurs on antibodies. This is false. This is incorrect because epitopes they occurs on the antigens, and the paratopes are occur on antibody. Epitopes are known as antigenic determinant. Paratopes are not. The paratopes are known as the antigen binding site. Okay, and this is the visual representation of the antigen antibodies, epitopes and paratopes. Okay, so this is the epitope and paratope binding. Okay, this is a epitope, it binds with the paratope, and the part of the antigen it is recognized by the antibody. Okay, so remember these things. Now I will go to the next problem. I hope there is no doubt. If there is doubt, please let me know in the chat box. Problem number 11, it says which of the following statement is incorrect? Your options are: number A, production of monoclonal antibody is expensive. Number B, monoclonal antibodies interact with the different types of epitopes. Number C, production of the polyclonal antibodies does not require hybridoma cell lines. And number D, monoclonal antibodies are used as the therapeutic drugs. Which of the following statements is correct? Number A, production of monoclonal antibody is expensive. Number B, monoclonal antibodies interact with the different types of epitopes. Number C, production of the polyclonal antibody does not require hybridoma cell lines. Number D, monoclonal antibodies are used as therapeutic drugs. Read all the options very carefully. Then let me know in the chat box A, B, C or D which of the following is the correct answer for problem number 11. Again, I will repeat which of the following statements is incorrect. Number A, production of monoclonal antibody is expensive. Number B, monoclonal antibodies interact with different types of epitopes. Number C, production of polyclonal antibodies does not require hybridoma cell lines. And number D, monoclonal antibodies are used as therapeutic drugs. Let me know in the chat box. Let me check if there are any answer for problem number 11. Okay, I got one answer which is saying it is number C. Let me check if number C is the correct answer for the problem number 11. Please let me know if there is any doubt in any question. Okay, feel free to ask if there is any issue with any question. So, problem number 11, number C is the given answer in the chat box. Now, let me check if number C is correct or not. So, the correct answer will be, it is number B. Monoclonal antibodies interact with different types of epitopes. This statement is incorrect. Okay. Now, what are the monoclonal and polyclonal antibodies? So, monoclonal antibodies are that are identical because they are produced by one type of immune cell or B cell, all clones of the single parent cell. And polyclonal antibody represent the polyclonal antibody. They represent they represent the antibodies from multiple clones of the B lymphocytes and therefore bind to a number of different epitopes. For example, immunoglobulin. Okay, so monoclonal antibodies are identical, but polyclonal antibodies are not because the monoclonal ones are produced by one type of immune cells. What are the now differences between the monoclonal antibodies and polyclonal antibodies? So, monoclonal antibodies refer to a homogeneous population of the antibodies that are produced by a single clone of the plasma B cell, and polyclonal antibodies refer to a mixture of the immunoglobulin molecules that are secreted against a particular antigen. Monoclonal antibodies are produced by the same clone of the plasma B cell and the production requires the hybridoma cell lines. But polyclonal antibodies produced by different clones of the plasma B cells and the production does not require hybridoma cell lines. Monoclonal antibodies a homogeneous antibody population. Polyclonal ones are a heterogeneous antibody population. Monoclonal antibodies interact with a particular epitope on the antigen, but polyclonal antibodies, they interact with different epitopes on the same antigen. Monoclonal antibody, the production is expensive. The polyclonal ones, the production is not expensive. The monoclonal antibody, the skills are required to handle the technology. Fewer skills are required for the production of the polyclonal antibody. The production of the monoclonal antibody takes some time. The production takes less time in case of polyclonal antibody. The monoclonal antibodies possesses less cross-reactivity and are used as therapeutic drugs, but the polyclonal antibody possesses comparatively high cross-reactivity and used as the general research applications. Advantages include immortal supply, high specificity and high reproducibility. This in case of the monoclonal antibody. In case of polyclonal antibody, the advantages include high affinity, tolerance of minor changes, and more robust detection. Okay, so these are the differences between the monoclonal antibodies and polyclonal antibodies. I hope it is clear. If there is any doubt, please let me know. Otherwise, I will proceed to the next problem. Okay, let me go to the next problem. If there is any, okay, let me check the chat box. So is it clear what is the polyclonal, what is the monoclonal antibody? If there is any doubt, please feel free to ask. Okay, let me go to the next problem then. If there is any there is no problem. Problem number 12 says, what is the use of the hybridoma technology? Your options are: number A, separation and isolation of the monoclonal antibody from a polyclonal set. Number B, separation and isolation of the lymphocytes from the immunoglobulins. Number C, separation and isolation of the haptens from the epitopes. Number D, separation and isolation of haptens from the enzymes. What is the use of the hybridoma technology? Again, I will repeat the options. Number A, separation and isolation of the monoclonal antibody from the polyclonal set. Number B, separation and isolation of the lymphocytes from the immunoglobulins. Number C, separation and isolation of the haptens from epitopes. Number D, separation and isolations of the haptens from enzymes. What is the use of the hybridoma technology? I will give you 1 to 2 minutes, then I will check the chat box and we will discuss about the hybridoma technology. What is the use of the hybridoma technology? Number A, separation and isolation of the monoclonal antibody from a polyclonal set. Number B, separation and isolation of lymphocytes from immunoglobulins. Number C, separation and isolation of haptens from epitopes. And number D, separation and isolation of the haptens from enzymes. Let me check the chat box. So is there any answer for problem number 12? I got one answer which says it is number A. Okay, let me check if number A is the correct answer or not for the hybridoma technology. Let me now check. So, what is the use of hybridoma technology? The correct answer will be very good. Number A, separation and isolation of monoclonal antibody from a polyclonal set. So basically, hybridoma technology is a method that is used to produce the large quantities of identical antibodies, which are known as the monoclonal antibodies, by fusing the antibody producing B cells with the myeloma cells. Okay, so this is a method of producing. This was discovered by the Kohler and Milstein in 1975. They were awarded Nobel Prize in Physiology and Medicine in 1975 for this for this discovery. The hybrid cells, which are produced by fusing the B lymphocyte with myeloma cell or tumor cells, are known as the hybridoma cells. Okay, the B lymphocyte have the ability to produce large number of antibodies, and tumor cells in indefinite had the indefinite growth. That is why two cells used to production of the hybrid cell. So this is a monoclonal antibody reacts with a specific epitope on the given antigen. This is a polyclonal antibody which reacts with the various epitopes on the given antigen. So what happens? We have the myeloma cells, okay, which have the power to differentiate in the indefinite growth, and these are the B cells or B lymphocytes, which have ability to produce large number of antibodies. So both are fused, and the hybridoma cells, the fusion of B lymphocyte and myeloma cells give us the hybridoma cells. This the culture, then test is happened, and the screening of the hybridoma cells happen. Then hybridoma they will propagation and positive clone, and the in vitro and in vivo propagation, this will lead us the monoclonal antibodies. Okay, so that is how the production of monoclonal antibody happens by using the hybridoma technology. Okay, is there any doubt? You can ask me. Okay, otherwise I will move to the next problem. So this is all about the hybridoma technology. You have to just understand what is it, and just you have to remember what is the rationale behind this technology. Now let me go to the next problem. Problem number 13. It says that which of the following statements is true regarding the antibodies? Your options are number A, B cells are associated with the cellular immunity and T cells are associated with the humoral immunity. Number B, T cells help the lymphocytes to produce antigen. Number C, T lymphocytes differentiate into the plasma cells. Number D, B lymphocytes undergo maturation in bone marrow. A, B, C or D, which of the following statements is true regarding antibodies? Number A, B cells are associated with the cellular immunity, whereas T cells are associated with the humoral immunity. Number B, T cells help the lymphocytes produce antigen, and number C, T lymphocytes differentiate into the plasma cells, and number D, B lymphocyte undergo the maturation in bone marrow. Which of the following statements is true regarding the antibodies? Let me know in the chat box A, B, C or D, which of the following you think is the correct answer. I will wait for 2 minutes, 1 to 2 minutes, then I will check the chat box. Read the options very carefully and then let me know. Which of the following statements is true regarding the antibodies? Let me check the chat box. Let me check if there are any answers. Okay, I got one answer which is saying it is number C. Let me check if number C is the correct answer or not. Okay, the statement which is true regarding the antibodies will be number D. B lymphocytes undergo maturation in bone marrow. This statement is true. Now let us discuss what is an antibody, a blood protein produced in response to and counterattacking a specific antigen Antibodies combine chemically with the substances which is the body recognized as aliens, such as bacteria, viruses, and foreign substances in the blood. Antibodies, also known as the immunoglobulin, are Y-shaped proteins that is produced by the immune system to help to stop the intruders from harming the body. And now when the intruder enters the body, immune system springs into the action. These invaders, which is called the antigens, can be viruses, bacteria, or other chemical. Now, when an antigen is found in the body, the immune system will create antibodies to mark the antigen for the body to destroy. So B cells, what does they do? They attack invaders outside the cell. T cell, they attack the infected cells. So what are the differences between the T cells and B cells? T cells originate in bone marrow, mature in thymus. B cells originate in and mature in bone marrow. Mature T cells occur inside the lymph node. Mature B cells occur outside the lymph node. T cells, they bear the TCR receptor. B cells bear the BCR receptor. T cells recognize viral antigen on the outside of the infected cells. B cells recognize antigens on the surface of the bacteria and viruses. T cells have longer lifespan than B cell. B cells, T cell, they lack surface antigen. B cells have surface antigen. T cells secrete lymphokines and are involved in the state cell mediated immunity. B cells, they secrete antibodies and are involved in the humoral or antibody mediated immunity. 80% of the blood lymphocytes are T cell, 20% of blood lymphocytes are B cell. They have three types of helper T cells, cytotoxic T cells and suppressor T cell. B cells are of two types, plasma cells and memory cells. T cells move to the site of the infection and act against the tumor cells or transplants. B cell does not move to the site of the infection and does not act against the tumor cells or transplants. T cells have the inhibitory effect on the immune system. B cells, they does not have the inhibitory effect on the immune system. T cells defend against the pathogen including virus, protease, fungi and enter the cells in the body. B cells defend the bacteria and viruses in the bloodstream and the lymph nodes. Okay. So these are the differences between the T cells and B cells. Each and every points are very important and you have to remember these things because these antibodies, T cells, B cells are very much important. Now I will go to the next problem. Problem number 14. Which of the following statement is false? Number A. Both receptors and enzyme have active site. Number B. Only enzymes have active site, not the receptors. Number C. After binding, reaction takes place in the enzymes. Number D. After binding, no reaction takes place in receptor. Which of the following statement is false? Again I will repeat. Number A, both receptors and enzymes have active site. Number B, only enzymes have active site, not the receptors. Number C, after binding, reaction takes place in enzyme. Number D, after binding, no reaction takes place in receptor. I will wait for 1 to 2 minutes again and then I will check the chat box. Meanwhile, try to answer the question. Which of the following statement is false? Number A, both receptors and enzymes have active site. Number B, only the enzyme have active site, not the receptors. Number C, after binding, reaction takes place in enzymes. Number D, after binding, no reaction takes place in receptors. Let me check the chat box. So is there any answer for problem number 14? Yes, I got a one answer which is saying it is number A. Let me check if number A is the correct answer or not. So the statement which is false will be Number B, only enzymes have the active site, not receptors. This statement is false. Okay, both enzymes and receptors have the active site. After binding, reaction takes place in enzyme, but no reaction takes place in receptor. So what are the receptor? Receptor is a protein molecule usually found embedded within the plasma membrane surface of the cell that receives the chemical signals outside from outside the cell, and when such chemical signals bind to the receptor, they cause some form of cellular and tissue response. Okay, so this is the receptor bind ligand receptor ligand binding. This is the receptor. These are the ligand, which are the primary messenger. When the ligands are bind to the receptor, it will there will be a signal transduction where the secondary messenger and cellular response will happen, and changes in gene expression will also happen. Okay, that is how the receptor actually works, by the signal transferring, and the binding of the ligand to the receptor will not cause any kind of chemical reaction, but it will cause a kind of the chemical the message transfer type sort of thing. So most receptors are membrane-bound protein that contain an external binding site for hormones and neurotransmitters. Binding results in an induced fit that changes the receptor conformation. This triggers a series of events that ultimately results in the change in the cellular chemistry. Neurotransmitters and hormones do not undergo a reaction when they bind to the receptors. They depart by the binding site unchanged once they are past their message. The interaction that bind a chemical messenger to the binding site must be strong enough to allow the chemical message to be received, but weak enough to allow the messenger to depart. Binding groups are the functional groups present in the messenger molecules, which are used for the binding to the receptor binding site. Binding regions are the regions of the receptor binding site which contain the functional groups capable of forming the intermolecular bonds to the binding groups of the messenger molecule. Okay, so this is about the receptors. Now I will go to the next problem. Problem number 15 says, which of the following agents activates a receptor to produce a submaximal effect but antagonizes the action of a full agonist? Your options are number A agonist, number B antagonist, number C partial agonist, number D inverse agonist. Which of the following agents activates the receptor to produce a submaximal effect but antagonizes the action of a full agonist? A, B, C or D, let me know in the chat box which of the following will be the correct answer for problem number 15. Which of the following agents, what is the correct answer for problem number 15? Let me check the chat box. Okay, I got one answer which says it is the number D. Let me check if number D is the correct answer or not for problem number 15. Okay. So, the correct answer will be number C, partial agonist. So, the following terms are used to describe the drug receptor interaction: agonist, an agent that activates the receptor to produce an effect similar to that of the physiological signal molecule; inverse agonist, an agent that activates the receptor to produce an effect that is opposite direction of the agonist; antagonist, an agent that will prevent the action of agonist on a receptor or subsequent response, but it does not have any effects of its own; Partial agonist, an agent that activates the receptor to produce the submaximal effect but antagonizes the action of the full agonist. And ligand is any molecule that attached to selectively to the particular receptor or site. The term only indicates the affinity or ability to bind without the regard of the functional change. Agonist, competitive antagonist are both ligands of the same receptor. So this is a very good graph actually, which describes the effect of this this particular agents. So basically, this is our receptor, this have the drug, and the drugs can be a full agonist, a partial agonist, a inverse agonist, or an antagonist to a particular receptor. Okay, so if it is a, so this is in the this graph, in the y-axis we have the effect, in the x-axis we have the log dose. So what happens in case of the agonist, we have a full effect, as you can see the curve. The inverse agonist, it forms the similar effect but in opposite direction. Okay, so inverse agonist which activates the receptor and produced an effect which is exactly opposite to a full agonist. Partial agonist which produces an submaximal effect, okay, which is not high like the full agonist, but there is some effect. Antagonist which does not produce any effect. So as you can see, it is a straight line parallel to the x-axis because there is no effect. Okay, now this graph actually explains very well about the effects of this agonist, of inverse agonist, partial agonist, and antagonist. Okay, now I will go to the next problem. Problem number 16. So this is a mathematical problem. Okay, the turnover number of the chymotrypsin is 100 per second, and for DNA polymerase it is 15 per second. The velocities of the reaction catalyzed by both enzymes at the saturated substrate levels can be made equal if dash times more than DNA polymerase, the dash times more DNA polymerase than chymotrypsin were used. Fill in the blanks. Your options are number A 1500, number B 0.15, number C 67, and number D 6.7. Again, I will repeat, the turnover number for chymotrypsin is 100 per second, and for DNA polymerase it is 15 per second. The velocities of the reaction catalyzed by both enzymes at saturated substrate levels can be made equal if dash times more DNA polymerase than chymotrypsin were used. Fill in the blanks. For this, I will wait for 2 minutes. Try to solve this problem. You have to use the pen and paper to for solving this kind of questions, but this type of mathematical questions are very crucial and it may come to your examinations also, so please try. I will give you 2 minutes and try if you can solve this question, otherwise I will show you how to solve it, but first you have to try. So what will be the answer? Let me check the chat box. Okay, there is no answer. Okay, let me show you how to solve this kind of problem. This kind of mathematical problems will actually come to your examination also. So you have to prepare accordingly. So the correct answer will be number D 6.7. Now let me show, first let us discuss what is the turnover number. So basically turnover number or K cat is the The unit is second inverse, is defined as the number of product molecules formed by the each enzyme active site per second. Okay, the equation goes like this: Vm or Vmax equal to Kcat into E total. E total means the total concentration of the enzyme, and the Kcat is the turnover number, Vm is the maximum velocity. Now think as Kcat as the frequency of catalysis, the number of catalytic cycles per second. The value of the Kcat is the measure of the catalytic efficiency. Faster enzyme have the highest Kcat values. Only Kcat is a true constant. Its value only depends on the enzyme's catalytic power. Note that Vm cannot be a true constant because it also depends on the the concentration of the enzyme. Now Michaelis-Menten equation goes like this: V0 equal to K2 S0 by E0 divided by S plus Km. At the high substrate concentration, when the substrate is much greater than Km, the equation looks like this. Okay, rate equal to, or Vmax equal to K2 by E0. That is the rate attains the maximum value when the reaction is the zero order with respect to the substrate concentration. This K2 is called the turnover number, which represents the number of molecule converts to the product by unit time, 1 unit per enzyme. Let us assume Xc is the strength of the molarity of chymotrypsin enzyme and Xd is that of the DNA polymerase. So if we replace these things in the equation, we will get Vmax of chymotrypsin equal to 100, which is the turnover number for the chymotrypsin, into the concentration of chymotrypsin, and the Vmax for DNA polymerase will be 15, which is the turnover number for the DNA polymerase, into Xd, which is the concentration of the DNA polymerase. If the Vmaxes are equal, so we will get the this, if means these two equations are equal, which means that 100 into Xc, which is the 100 into the concentration of chymotrypsin enzyme, equal to 15 into the concentration of DNA polymerase. Then Xd by Xc, which is the will be the 100 by 15, which is the 6.7, means the concentration of DNA polymerase has to be 6.7 times more than that of the chymotrypsin. Okay, when Xd equal to 6.7 into Xc, then these two velocities will be equal. That is the question that it has asked. Okay, it might seem like a little difficult, but once I shared this PPT, you can try it by yourself, and then you will find it is a very easy actually to solve. Just you have to write the things, put all the things are given in the question itself. Just you have to put the things in a rational manner. Okay. Try. If you do not understand, let me know via email, or in the beginning of the next class I will repeat this question again. Every mathematical question, after I share the PPT, please try by yourself. If you have difficulty in solving, let me know before the next class. I will definitely repeat that in the next class also. Now let us go to the next problem. Problem number 17 says, which inhibition has the apparent Km value as the Km value of the normal enzyme catalyst reaction? Your options are number A competitive, number B non-competitive, number C uncompetitive, and number D mixed. I think I have discussed it in my previous one slide. I think I have given you a hint, or I might have discussed it itself. So basically, when we are using there, when there is no inhibitor in Michaelis-Menten equation, we have a Km value, we have a Vmax value, right? But in presence of the inhibition or inhibitor, this Km and Vmax value changes from the from the normal enzyme catalyst reaction. These are known as the apparent Km and apparent Vmax. You have to identify which inhibition has apparent Km value, same as the normal enzyme catalyst reaction. Let me know in the chat box. I will wait for a minute, then I will show you the correct answer. I will wait for 2 minutes, so 1 or 2 minutes, then I will show you the answer and we will discuss it. Which inhibition has the apparent Km value same as the normal value of the normal enzyme catalyzed reaction? Let me check the chat box. Number D, I got one answer which says it is number D. Now let me check if number D is the correct answer or not. So the correct answer will be non-competitive inhibition. I think I have shown you it in one slide where I have just mentioned the graphs. I think I have mentioned it earlier also. So when we were discussing the graph here, I have mentioned that in case of non-competitive inhibition, they both lines are actually meets on the same point on negative y-axis, which makes the Km same. Okay. So this is the same thing. If you remember the graph, you can answer the thing. Okay, anyways, you can remember this thing by remembering the chart. Okay, so this is a little bit difficult. It may seem a little difficult. You don't have to memorize all the thing, but if you can remember, it is better. Now, all these things can be derived from the steady state equations, which I have mentioned earlier. Okay. So in case of competitive inhibition, the apparent Vmax is same as the normal Vmax. The value of apparent Km is like this: Km equal to 1 plus concentration of I by Ki, where concentration of I is the concentration of inhibitor. In case of mixed inhibition, this is the thing: Vmax divided by 1 plus I by K. So these are the things I am not going to read out. You can just check it later also. So all these things actually come from the steady state approximation of the all the inhibitions, enzyme inhibitions, which I have already mentioned. Okay. But the derivations are not included in your syllabus. That is why I am not going to discuss the derivations. But I thought it is better to give you a overview that the Vmax and Km from the normal enzyme catalyzed reactions are not same as the same as the Km and Vmax of the in presence of an inhibitor. Okay. Now let me go to the next problem. Problem number 18 is also a mathematical question. It says consider the following plot of 1 by V0 versus 1 by S plot in the Lineweaver bark. The results obtained from the graph is number 1, intercept value is 0.2 micromolar per minute inverse and the slope is 7 into 10 to the power 5 minute. What is the Km value for the reaction? Your options are number A 35 into 10 to the power 5 micromolar, number B 0.258 into 10 to the power 5 molar, number C 7 into 10 to the power 6 micromolar and number D 0.011 molar. What is the Km value for the reaction? The 1 by V0, 1 by S plot, the intercept and slope values are given. It is very easy. I have mentioned it, number of time, the Lineweaver bark plot. What is the slope, what is the intercept? Just you have to put the values in those things and you will get the answer. Consider the following values. Let me know. Okay, let me check the chat box. We don't have much time, but we have to solve a couple of mathematical problems before going. Okay, there is no answer. Let me show you how to solve this kind of problem. See, in case of the Lineweaver bark plot, I have, so correct answer will be number A, 35 into 10 to the power 5. Now, how to do this thing? I have mentioned it couple of times actually. So, in Lineweaver bark plot, the Lineweaver bark plot is a double reciprocal plot, is a common way of illustrating the kinetic data. This is produced by taking the reciprocal from both sides of the Michaelis-Menten equation. This linear form of Michaelis-Menten equation produces a straight line with the equation y equal to mx plus c, where y-intercept is equivalent with 1 by Vmax and x-intercept is the graph represented by 1 minus 1 by Km. Double reciprocal form of the equation is this: 1 by V equal to Km by Vmax into S plus 1 by Vmax, where y is this, y into mx, m is the Km by Vmax is the slope and intercept is 1 by Vmax. So accordingly, the question intercept is 1 by Vmax, slope is Km by Vmax. Now the question has given that the slope intercept value 1 by Vmax value is 0.2 micromolar minute inverse. So Vmax will be So Vmax being 1 divided by 0.2 means 5 micromolar per minute, and slope is Km by Vmax. So the slope value is Km by Vmax. You know the Vmax, you know the slope value. Now you will know what is the Km. So Km will be slope into Vmax. The slope given is 7 into 10 to the power 5 minute, and the Vmax is given 5 micromolar per minute. So now what is the value of Km? Km is equal to 7 into 10 to the power 5 into 5 means 35 into 10 to the power 5 micromolar. Minute cancel, minute and per minute, these are cancelled out. So the Km is 35 into 10 to the power 5 micromolar. So that is how you have to solve this type of question. See, whatever the question comes from the line viewer, bark plot and all these things, it will be related to your slope and intercept value. If you know what is the equation, if you know what is the slope, what is the intercept value, then easily you can solve this type of question. Okay. Anyways, I have already mentioned that the mathematical question, if you have any doubt, you can let me know in the next class also. I will discuss these questions again in next class. It is understandable, right? Do you have any problem? If there is no problem, I will go to the next question. Okay, I think it is clear to all. Then I will move forward to the next. This is very easy actually. This kind of Michael line weaver bark plot related questions are very easy because you only have to remember what is the slope and what is the intercept. Okay. Now let us go to the next problem. Problem number 19. It says that consider the plot of the S divided by V0 versus S. Okay, of the Hannes wolf. The results obtained of the graph is the intercept value 0.5, slope value 0.005. What is the Km value? Your options are number A 5000 millimolar, number B 1000 millimolar, number C 2500 millimolar, and number D 100 millimolar. Again, one more graph related question is given. This you can also derive from the Hannes-Wolff plot, can be also derived from the Lineweaver-Barth plot itself. It is very easy. Try to solve it. I will give you 2 minutes. Let me check the chat box if there is any answer. Okay, there is no answer. Let me show you how to solve this kind of question. See, this is the correct answer is number B, 1000 micromolar millimolar. So Hansable Prof is nothing but, so we know what is the what is the our Lineweaver bark plot. Lineweaver bark plot is this one by V equal to Km by Vmax into S plus 1 by Vmax. Only what you have to do is multiply the Lineweaver bark plot with the substrate concentration. That will give you the Hans-Wolff plot. See, Lineweaver bark plot and Hans-Wolff plot, only the difference is it is just multiplying the Lineweaver bark plot with substrate concentration will gives us the Hans-Wolff plot. This is the one y-axis, we have a substrate concentration divided by V, and on the x-axis we have substrate concentration. The slope is 1 by Vmax and the intercept is Km by Vmax. Okay. So now Hans-Wolff plot is associated with this equation. So plotting this intercept is Km by Vmax and slope is 1 by Vmax. According to the question, Km by Vmax is 0.25 and okay, sorry. So there is a mistake in the printing. Okay, so there is a mistake in here. Okay, sorry, here is a mistake in here. Okay, yeah. So, so the question Km by Vmax equal to 0.5, right? Km by Vmax is 0.5 and 1 by Vmax is 0.005. So Vmax is 2000 millimolar if you just make it reciprocal, and Km value is 0.5 into 2000 is 1000 micromolar. Okay. So is it clear or is there any doubt? You can let me know if there is any doubt. Is there any doubt? You can let me know. Nothing. Hence rule plot is nothing. You just have to make the reciprocal of our particular your you have to just multiply our this Lineweaver bark plot with substrate concentration. Then you will get this thing. According to the question, Km by Vmax is the intercept which is 0.5 and 1 by Vmax is the slope is 0.005. So Vmax will be 2000 millimolar and Km will be Vmax into 0.5 means 1000 millimolar. Okay. If there is no doubt, I will move to the last question of today's session. Okay, there is no doubt. Let me go to the last question of today's session then. So the last question of today's session, it says that consider the following values of Michaelis-Menten enzyme kinetics and calculate the dissociation constant for the enzyme substrate complex and the catalytic efficiency of the enzyme. K1 equal to 4 into 10 to the power 8 molar per minute inverse. K minus 1 equal to 1 into 10 to the power 3 minute inverse. K2 equal to 6 into 10 to the power 3 minute inverse. Your options are number is 0.5 micromolar and 0.343 into 10 to the power 8 molar per minute inverse. Number B is this 2.5 micromolar and 8.57 into 10 to the power 8 molar per minute inverse. Number C 2.5 micromolar and 3.45 10 to the power 8 molar per minute inverse. Number D 5 micromolar and 0.345 10 to the power 8 mole per minute inverse. You have to identify what is the dissociation constant and what is the catalytic efficiency of the enzyme. I will give you some time. I will show you how to solve this type of question. You can practice it by yourself. The mathematical questions, this is one is little bit tricky, but the others were very easy. The questions which are derived from the graph, the Michaelis, the Lineweaver bark plot, and also the Hansville plot. Since we don't have much time, let me show you how to solve this type of problem. Let me first check the chat box. I think there is no answer, but let me just confirm. Okay, let me just show you how to solve this kind of question. This kind of question, this might be a little tricky, but it is not that hard. It is easy also. Correct answer is number C. Let me show you. So this you have to remember. Okay, the dissociation constant from the enzyme substrate complex is given by K minus 1 by K1. And the catalytic efficiency is given by K2 by Km. This you have to remember. The formula you have to remember. Okay. So the dissociation constant is K minus 1 by K1. K minus 1 is given 1 into 10 to the power 3 minute inverse. Okay. And K1 is 4 into 10 to the power 8 molar minute inverse. Molar inverse, minute inverse. If you divide these things, you will find the answer. Okay. So the dissociation constant will be 0.25 into 10 to the power minus 5 molar, which is 2.5 micromolar. And catalytic efficiency is K2 by Km. Now Km, Michaelis-Menten constant, is given by K minus 1 plus K2 by Km by K1, right? So it is goes like this. K minus 1 is 1 into 10 to the power 3 plus 6 into 10 to the power 3, which gives us the 7 into 10 to the power 3 divided by K1, which is 4 into 10 to the power 8. The minute inverse are cancelled out. We have only molar. So the value is 1.75 into 10 to the power minus 5 molar. This is Km. Now catalytic efficiency is K2 by Km. K2 is 6 into 10 to the power 3 minute inverse and divided by 1.75 into 10 to the power minus 5 molar. So you get this value 3.43 into 10 to the power 3 molar plus minute inverse. So again, what I am saying, that mathematical questions, these are very simple, okay, but sometimes it get tricky. You just need to remember what is the formula. You have to put the particular values. The all the things will be given in the question itself. You have to just put the values into the formulas. If you do not remember the formula, sometimes you can derive it. Just in case of the graphical questions like from the Lineweaver, Burke and Hansbull plot, you can derive the slope, what is the slope, what is the intercept. If you just, you have to know what is the Michaelis-Menten equation for those things. This type of questions might be a little tricky, but it is not difficult at all. You just have to remember what is the values or what are the formulas. So this was the last question of today's session. So today what we have done, we have discussed about the different types of the enzyme inhibition. We have discussed, and let me first share my email ID. So this is my email ID. Now, if you have any doubt, any query regarding today's class, regarding any previous lecture or any topic which we have covered till now, please let me know via this email ID. Okay, I will answer your queries via email, and if it is not possible to do it by email, I will discuss in the next classes. So today we discussed, I mean, every type of enzyme inhibition we have discussed. We have discussed lots of examples of different enzyme inhibitions, and we have solved a number of mathematical questions also. This PPT and this class recording I will be sharing to the SWAYAM website, which you can, I have already mentioned where to find in the beginning. You can go to the problem solving session on the left panel, and you can download the recording and the PPT from that position. Okay. Mathematical questions, please try to practice one or two times. I think you will be fine if you practice for a lot of time. I will close the session for today if there is no doubt. Thank you all for joining. Today it was our week 4 live session for this course of organic chemistry and biology and drug development. We will meet on next Friday for our week 5 live session. Okay. Till then, goodbye, and please feel free to reach me through email if you have any doubt. I am closing the session for all. Thank you for joining and thank you for answering also.